168, and Mir-159 from vector control in STTM 165-166-48 were calculated. Figure 3c. Although the read numbers of both Mir-168 and Mir-159 were nearly at the same level between vector control in STTM-165-166-48, the reads of Mir-165 and Mir-166 were dramatically reduced in STTM 165-166-48 compared with vector control. This further demonstrates that STTM triggers a drastic and highly specific reduction of the targeted small RNAs in vivo. Contribution of spacer length and STTM expression level to STTM efficacy. Both STTM 165-166-31 and STTM 165-166-48 independent transgenic lines displayed variable phenotypes. These phenotypic variations, likely due to positional or transgene copy effects, allowed us to examine the potential relationship among the phenotypic severity, the STTM transcript level, and the degree of reduction in Mir-166 levels in independent transgenic plants. For each population of the STTM 165-166-31 and the STTM 165-166-48 transgenic plants, we analyzed two types of independent transgenic lines with either weak or strong phenotypes. For either STTM 165-166-31 or STTM 165-166-48, much higher levels of STTM transcripts and significantly lower levels of Mir-166 were detected in transgenic plants displaying severe phenotypic defects than in transgenic plants displaying less severe phenotypes. Figures 4a and 4b. This indicates that for both 31 nucleotide spacer and 48 nucleotide spacer STTMs, the expression level of STTMs plays critical roles in the downregulation of the targeted mRNA. The higher the expression of the STTM, the more the reduction in mRNA levels and the more severe the phenotypic alterations. Comparisons of STTM and Mir-166 levels in STTM 165-166 transformants with different spacer lengths. A. qRT-PCR analysis of STTM and Mir-166 levels in independent STTM 165-166-31 transgenic plants. Bars show say. B. qRT-PCR analysis of STTM and Mir-166 levels in independent STTM 165-166-48 transgenic plants. Bars show say. C. Comparison of STTM and Mir-166 levels between independent STTM 165-166-31 transgenic plants and independent STTM 165-166-48 transgenic plants. Transgenic plants used in A2C were independent lines. Actin mRNA, for STTM, or SNO-R101, for mRNA, was used as an internal control. Values were obtained by normalizing to actin or SNO-R101 and then comparing the normalized values to those of control plants. Different shades of gray are used to indicate different independent lines to allow for the easy comparison between STTM and Mir-165-166 levels in the same lines. Bars show say. Note: the vector controls also contain 2xp35s and T35s. See supplemental figure 7 online, which bind the common qRT-PCR primers for the quantification of STTM expression and thus gave a value by qRT-PCR. We further compared the efficacy of the 31 nucleotide spacer and the 48 nucleotide spacer STTMs in the reduction of Mir-166 levels. Although in some cases the STTM expression level in STTM 165-166-31 transgenic plants was higher than that in STTM 165-166-48 transgenic plants, STTM 165-166-31 was not as effective as STTM 165-166-48 in triggering the reduction of Mir-166 levels, figure 4c. Together, these data demonstrated that both the expression level of STTM and the length of the linker inside the STTM are important in the downregulation of mRNAs in Arabidopsis. STTM efficacy requires both the spacer and the two mRNA binding sites. Compared with the IPS1-based TM, a featured distinction of STTM is that it contains one spacer and two target mRNA binding sites. To further evaluate the contribution of the spacer and the two binding sites to the efficiency of STTM, a set of STTMs was engineered to have different variations in their structures based in the STTM 165del, 166-48 structure. First, we examined the effects of STTM 165del, 166-48 and STTM 165-166del 48 constructs, Figure 5a, in which we kept one binding site of Mir 165 or Mir 166 and deleted the spacer in the other binding site in the STTM 165-166-48. Both kinds of transgenic plants showed reduced levels of Mir 165-166, Figure 5c, but displayed no obvious phenotypic alterations, Figure 5b. Therefore, constructs containing one mRNA binding site were not as effective as STTMs in blocking the function of mRNAs, even when expressed by a 2x35S promoter. Comparison of the efficacy of STTM constructs with one mRNA binding site. A. Diagram of STTM with one Mir 165 binding site or one Mir 166 binding site. Blue indicates the bulge sequences in the mRNA binding sites. B. Phenotypes of transgenic plants containing STTM constructs with one Mir 165 binding site or one Mir 166 binding site. Bars equals 1.0 centimeters. C. RNA gel blotting to determine the levels of Mir 165-166 in transgenic plants containing STTM with one Mir 165 binding site or one Mir 166 binding site. Total RNAs were prepared from multiple primary transformants. We generated two additional constructs, STTM 165-MUT, 166-48 and STTM 165-166-MUT48, by mutating one mRNA binding site of STTM 165-166-48 without changing the overall length of the STTM and the spacer. In both cases, Mir 165-166 was expected to interact only with the non-mutated mRNA binding site, Figure 6a. In contrast with the STTM 165del, 166-48 and STTM 165-166del 48, both STTM 165-MUT, 166-48 and STTM 165-166-MUT 48 transgenic plants displayed obvious morphological defects, Figure 6b, but the phenotypes were not as severe as those observed in the STTM 165-166-48 transgenic plants. Comparison of the efficacy of STTM with one mutated binding site. A. Diagram of STTM with mutated Mir 165 binding site or mutated Mir 166 binding site. Orange indicates the spacer region. Blue indicates the bulge sequences in the mRNA binding sites. Red indicates mutations in the mRNA binding sites. B. Phenotypes of transgenic plants containing STTM with mutated Mir 165 binding site or mutated Mir 166 binding site. Bars equals 1.0 centimeters. C. RNA gel blotting to determine the levels of Mir 165-166 in transgenic plants containing STTM constructs with mutated Mir 165 binding site or mutated Mir 166 binding site. Total RNAs were prepared from multiple primary transformants. To gain more insights into the relationship between STTM structure and function, we analyzed the effects of two additional constructs, STTM 165-165-48 and STTM 166-166-48, Figure 7a, which have two identical Mir 165 or Mir 166 binding sites rather than one binding to Mir 165 and the other to Mir 166. We found that these STTMs resulted in strong phenotypes with drastically decreased Mir 165-166 accumulation, Figures 7b and 7c, which resembled those of STTM 165-166-48. Comparison of the efficacy of STTMs with two identical binding sites. A. Diagram of STTMs with two Mir 165 binding sites or two Mir 166 binding sites. Orange indicates the spacer region. Blue indicates the bulge sequences in the mRNA binding sites. B. Phenotypes of transgenic plants containing STTMs with two Mir 165 binding sites or two Mir 166 binding sites. Bars equals 1.0 centimeters. C. RNA gel blotting to determine the levels of miR-165-166 in transgenic plants containing STTMs with 2 miR-165 binding sites or 2 miR-166 binding sites. Total RNAs were prepared from multiple primary transformants. We further compared the efficacy of these different STTM constructs in the reduction of miR-166 levels. We found that although the expression level of STTM in STTM 165-165-48 or STTM 165-166-48 transgenic plants is slightly lower than that in the STTM 165-166 del48 or STTM 165-166 SMUT48 transgenic plants in some cases, the extent of reduction of miR-166 is higher. See supplemental figure 6 online. In summary, among many STTM 165-166 del48 or STTM 165 del 166-48 transgenic plants, none of them displayed obvious abnormal phenotypes. In the case of STTM 165 MUT 166-48 or STTM 165-166 SMUT48 transgenic plants, approximately 20% of the transformants displayed abnormal phenotypes, but the phenotypes were not comparable to the strong abnormal phenotypes of STTM 165-165-48 or STTM 165-166-48 transgenic plants. Overall, approximately 30% of STTM 165-165-48 or STTM 165-166-48 transformants displayed strong abnormal phenotypes. These data are in agreement with the expression levels of miR-166, the corresponding levels of STTM transcripts, and the contributions from the spacer and the mRNA binding sites in each category of modified STTM constructs. By dissecting and comparing the effects of differently modified STTM constructs, we conclude that both the spacer and the two mRNA binding sites are necessary for the effectiveness of STTM in the functional blockage of small RNAs. STTM directed functional blockage of other mRNAs and endogenous siRNAs. To evaluate the general applicability of STTM to the functional studies of mRNAs and siRNAs in plants, we use the STTM48 structure to target two additional mRNA families, miR-156-157 and miR-160, and one family of endogenous trans-acting siRNAs, tacrNAs, D7+, and D8+, in Arabidopsis, figure 8a. Similar to the miR-165-166 family composed of highly homologous but not identical mRNAs, miR-156 and miR-157 are different in only 3 nucleotides, figure 8a, and both target the squamosa promoter binding protein-like, SPL, transcription factor genes, including SPL3, SPL4, and SPL5, which are involved in the promotion of vegetative phase transitions as well as flowering. Wu and Pathig, 2006. Functional blockage of miR-156-157 is predicted to trigger an early